All right. So this week we're going to start talking about cloning. And so what is cloning? Well, basically, what we're going to do is we're going to take something from the flow of life, and we're going to cram it into a plasmid. And that is the foundations of cloning, right? Just taking some sort of genetic information and putting it somewhere else. But in particular, what we're going to do is take um, the messenger RNA for that encodes a protein, and we're going to put that into a fluorescent fusion plasmid so that we can express this protein of interest in as a fluorescent fusion protein. Okay, and so again, we're you can kind of consider it as like co-opting and forcing gene expression. Okay. And if you remember from last time, right, we get transcription from a genetic locus. Normally, it starts off as a really big pre-messenger RNA or a primary transcript that gets spliced. All the introns get cut out, and all the exons get spliced back together. And we're creating a much smaller piece of RNA than the primary transcript. There's also the capping and polydenylation processes that happen at the 5' and 3' regions specifically. Um, but what happens to that messenger RNA next? Okay, and so here's our representation of our spliced messenger RNA again. And one of the things I pointed out last time was this AUG and this UGA in this particular thing. And that represents what's called the coding region of the messenger RNA, or it's also considered an open reading frame. Okay, so when a messenger RNA is decoded into protein, and that's shown in this little cartoon here, here's our messenger RNA. There are things called tRNAs that come in and recognize the bases on the messenger RNA. tRNA stands for transfer RNA. The tRNAs have an amino acid attached to them. And as the process of translation happens, the amino acids all get added one to the next, to the next, to the next, creating a chain of amino acids. That chain of amino acids is protein. But what's the actual decoding that happens, right? So we have four different nucleotides, A, T, C, or G. In RNA, it's not T, it's U, but same idea. Um, we have three different positions. Okay, so three consecutive nucleotides. So if we have four possibilities at three different positions, we have four possible ways to decode the messenger RNA. Or did I say 64? 64 possible ways to decode the messenger RNA. There's only 20 amino acids that get encoded from messenger RNA. So there is some redundancy among the codons, okay? But the one thing you should always think about when you're looking at a messenger RNA sequence and thinking about how it gets decoded into protein, we have to look for the start codon, okay? AUG, or if you're looking at the DNA sequences like we're going to be looking at this week, ATG. Um, it's the same idea. AUG, start codon. Okay, so burn this into your brains. This is a critical piece of information that you always need to think about. If someone hands you a messenger RNA sequence and says decode this thing, always look for the start codon. If there's not a start codon, 99.9% of the time that messenger RNA is not going to be decoded into protein. Okay. And so, the ribosome, which is shown here in blue, is the thing that actually facilitates the decoding. So the ribosome hops on at the beginning of the messenger RNA and starts scanning down until it finds a start codon. Then the tRNA for that start codon, that base pairs with that start codon, hops on and starts decoding the messenger RNA until it hits a stop codon. Okay, and there are three different stop codons. There's only one start codon. So our possibilities here are UAA, UAG, or UGA. Okay, so when the ribosome sees that, there's a terminator tRNA that pops in there and causes translation to stop. Okay, always one start codon, AUG. Learn that, memorize it, burn it into your brains. Three different stop codons. Um, Okay. And this is just a table to show you how the codons are decoded. So if we have GCA, GCC, GCG, GCU, any of those four encodes the amino acid alanine that has a single letter code of A. We have some like the start codon encodes methionine. AUG can also occur inside the messenger RNA. Um, um, so that one encodes methionine, but there's only one possibility for encoding methionine, just like there's only one possibility for encoding tryptophan, UGG. But you can see that across all of these amino acids that there is some redundancy. And so multiple different codons can encode the same amino acid, right? And so as the messenger RNA or as the ribosome decodes the messenger RNA, it goes 3 nucleotides, then it sees the next 3 nucleotides, then it sees the next 3 nucleotides, and so on. Okay, and so that choice of which 3 amino acids is called the reading frame. The region that encodes where the codons all line up, where you get an AUG to a stop codon, that's called an open reading frame, or the coding region of the messenger RNA. Okay, so when we clone a gene, it's really this part that we care about, right? For if we're trying to force protein expression, ultimately it's really the open reading frame that we care about. There are other times where we might care about the 5' or 3' untranslated region, but for the purposes of encoding a protein, it's really this open reading frame at the coding region that we care about, and that's ultimately what we're going to be cloning. Okay, so how do we clone this thing? Well, we've mentioned before that there's this region in the plasmid called the multiple cloning site, right here. And so this region has these specific restriction enzyme sites available to it. And these restriction sites are only present in the multiple cloning site. And so we can use that to our advantage and put things in using those restriction sites. And just a reminder, right, we have this promoter up here that's going to drive expression of whatever is in this multiple cloning site. And if we clone this properly, if we maintain our reading frames between our protein of interest and our GFP, we can create a fusion protein. And so wherever that protein of interest goes, the GFP is going to be attached to it, and the GFP will go around with it, and we'll be able to see that protein being expressed inside of the cells. Okay, so what about these restriction enzymes do we care about? Well, if you think about how we described them before, right, we have what's called a sticky end for most restriction sites. Okay, so here's our EcoR1, GAA-DTC. When the EcoR1 cuts the DNA, it cuts between the G and the A on both strands. And so on this strand, this gets cut like this. Sorry, this thing's annoying. It leaves these single base regions here, right? The AATTC on this side and the AATTC going this direction. And so those are single stranded pieces of DNA. And theoretically, since they're single stranded and they're complementary to one another, they can base pair with one another, okay? And so what we can do is when we design our primers for PCR, we can add restriction sites to them, okay? So, remember, when a primer binds, the tack is going to bind onto the 3' end and start copying there. What's at the 5' end of the primer doesn't necessarily mean much. As long as most of the primer can anneal to the template, right, towards the 3' end of it, it can still anneal. The tack can still hop on and make a DNA copy of our cDNA. So what we can do is do 2 different restriction enzymes, add 2 different restriction enzyme sites to our primers. And so by the time we make this PCR product, we're going to have a HIN3 site here and an AGE1 site on this side, okay? Some primer design considerations. One thing to think about is if we're making a fusion protein, do not include a stop codon, right? So we said that when the ribosome comes in and it sees a stop codon, it's going to pop off, okay? If we want this protein to be fused to a GFP, we want to make sure there's no stop codon. And so when we design our primers for this end, make sure that there's no stop codon being incorporated into there. We also need to make sure that we maintain the reading frame with our GFP. And so here, right, this ATG, what is that? Give you a second to think about it. ATG is the same as AUG. That's a start codon. Okay, so this is the beginning of the GFP. And so what this multiple cloning site diagram shows you is the codon usage. And we need to make sure that the codon usage of our open reading frame matches this particular codon usage, so that when our ribosome hops on here and starts decoding, eventually this thing is going to be put into the plasmid inside this region. And so it's going to get decoded along there, and eventually it's going to run into that GFP, and it's going to make one giant protein with a GFP on the end of it. Okay. One thing to take note of is that we're not going to use our regular old DreamTAC for this kind of PCR. We're going to use what's called fusion DNA polymerase. Okay, and this is a specially engineered DNA polymerase. It has this thing called a sliding clamp on it. And so we think, and we sort of misrepresent it, but enzymes sort of have an on and off rate. And so as we sort of represent this as like the TAC just hops on the DNA and starts copying. But in reality, it may copy 5 bases and then fall off, and a different TAC will hop on and copy 10 bases and then fall off and copy 6 more bases. And it's kind of random. And that kind of slows down TAC. And so what they've done is they've engineered this TAC to have a sliding clamp on it. And so it stays attached to the DNA as it copies. And that increases the speed of the copying a lot. This also increases the error rate, or the proofreading ability. So if we think about regular TAC, we get a mismatch. We get a wrong base incorporated about 1 in 228,000 base pairs. So it's going to be one wrong base all the time in there, in that 228,000 bases. For fusion, we get one error in 44 million bases. And so you can see this is a much cleaner PCR product, right? It's not going to have incorporated many errors. And so that's really important if we want to copy a gene and express it as a protein. We want to make sure we don't incorporate any errors as we do that. Okay, and so that's why we're going to use fusion here. We're going to use the same DNTPs, right? DNTPs are basically DNTPs, and so the free individual bases that get incorporated are still the same thing. But the fusion since it's a different enzyme, it requires a different buffer, right? Just how we use different buffers for our different restriction enzymes, the fusion polymerase requires a different buffer. And so make sure that you use the appropriate buffer for that one. So anyway, when we're done with our PCR, right? If our forward primer incorporated an EcoR1 site and our 3' primer or reverse primer incorporated a KPN site, this is a diagram of our PCR product. What we can do is then digest this DNA with those 2 restriction enzymes. We clean up our DNA to get rid of the enzyme, the restriction enzymes and the little DNA fragments that are, the cleanup process loses really small pieces of DNA and free nucleotides. And so what we're left with is a piece of DNA with 2 different sticky ends, right? So then if we take our plasmid DNA, and this is our PEGFPN1 here, we do the same digests on our plasmid DNA, and we have our complementary sticky ends here. And then we can join these 2 things together. Okay, so for this week, you're just going to be PCRing up your gene of interest. And again, it'll be whatever gene you amplified last week. And you're going to wind up cloning this ultimately, but we're going to start just by PCRing up our gene of interest. These are different primers than the primers you used last week. Okay, so the primers for last week were gene expression primers. We were just looking to see if the gene was expressed. Cloning primers have to incorporate these restriction sites into them. We also have to make sure that we're cloning the whole open reading frame and that we're not cloning the stop codon. Okay, and so this week I'm also going to have you design primers. And so you're only doing one actual PCR, but I'm gonna walk with you through primer design. So with all primers, you're going to need a forward and a reverse primer. Okay. And so when we're looking at a messenger RNA sequence, we're going to pull sequences out of that messenger RNA sequence that we decide are going to be good primer sequences. And when we find those, the forward primer is exactly the same as the sequence that we're looking at. But the reverse primer, we have to make complementary. So the reverse complement, right? So the matching base pairs going the opposite direction. So for our reverse primer, we always have to make sure to do the reverse complement. We're going to be working with messenger RNA sequences. So we're generally going to be sure that there's no introns present, but that'll help us when we're differentiating between a gene and gene expression. Okay. More primer design considerations. So when we design our forward and our reverse primers, we want to make sure that they're annealing temperatures. So remember, when we're going through our PCR cycle, we have our melting, our annealing, and our extension, right? That annealing temperature is when the primer sticks to the template. And that temperature is dependent on the base pair composition of the primer, right? And so we remember this DNA diagram that I've showed you now a bunch of times. When we look at A's and T's, there's only 2 hydrogen bonds there. And when we look at G's and C's, there's 3 hydrogen bonds there. So the more G's and C's that are present in a sequence, the higher your annealing temperature is going to be, because there's more hydrogen bonds that are holding that template to its the primer to its target sequence. If there's a lot of A's and T's, then it's going to be a weaker binding. And also the length of the primer is going to impact its annealing temperature. So generally the annealing temperature and melting temperature, I should mention, are essentially the same thing. It's just sort of whether you're thinking about them separating or you're thinking about them coming together. So TM means melting temperature, but when we say melting temperature and annealing temperature in this class, it's essentially the same thing. So the longer your piece of DNA is, the higher your annealing temperature is going to be. The more G's and C's, the higher your annealing temperature is going to be. And so when we're designing our forward and reverse primers, we want them to be within a degree. Ideally, within a half degree would be better. But sometimes you have to make some concessions. The other thing is that we try to have a G or a C at the 3' end of the primer. Okay, and so here's our 3' end down here. We want a G or a C down here in this 3' end because what's going to happen is the Taq has to hop on there and start incorporating bases. And the stronger this base pair is right here, the more stable that connection is going to be, and the easier it is for the Taq to start amplifying. If we have a bunch of A's and T's at the 3' end, then the 3' end of the primer can kind of fall apart a little bit, and it destabilizes that situation. And it makes it a little bit harder for the Taq to hop on it. It'll still work, but it's a little bit less reliable. And so I generally advise having G's or C's at the 3' end of the primer. Just consider that the primer is always incorporated into the new DNA strands, right? It's part of the PCR product that we create. The primer is not separate from the piece of DNA that we create. Primers generally should have a minimum of 16 nucleotides. This sort of balances the annealing temperature with the specificity of the primer. If your primer is too short, you can have some non-specific binding. If it's too long, you can also have some non-specific binding. 16 is a nice, happy intermediate. And again, make sure to order the reverse complement of the region that you design for your reverse primer. Okay, so this week you're going to be doing a fusion PCR with your gene of interest. Um, and so, whatever your gene was last time, make sure you get the cloning primers for that. Generally, the cloning primers are going to be labeled with the gene name plus GFP to indicate that um, a cloning primer that's going to be used as a GFP fusion. Um, excuse me. So you're going to run this fusion PCR. You're going to run 5 microliters of that on a gel. Um, so what you want to do is this fusion PCR is generally 50 microliters. So you're going to use 5 microliters to run in your gel. And if your gel looks good, then you're going to save that the rest of that PCR reaction. And we're going to use that next week. So make sure to label this really well and save it in the methods box so that we can use it next week. If it doesn't look good, then talk to me or the TAs, and we're going to ask you to rerun that PCR. We might change up the settings a little bit and attempt it again. Okay. Excuse me. Um, and finally, you're going to schedule a time to come do some primer design with me. The protocols for primer design are up there. You don't have to write down the dissolving oligonucleotides primer or oligonucleotides protocol. We're not actually going to do that. That's a good protocol to have in your back pocket if you ever want to design your own primers and order them and clone your own genes. It's a really useful protocol to have because when we order primers, they arrive desiccated, dry little pellets of DNA, and we have to rehydrate them. And so that protocol is useful for those situations. We're not actually doing that this week. All right, so that's it for this week.